Hello, everyone. Thank you for joining us this afternoon. Today, we have with us Mr. Sarath Dantu from Brunel University, London. Mr. Sarath did his master's and PhD in computational biophysics in the UK and holds a good experience as project manager with reputed organization. Currently, he is working as associate lecturer in Department of Computer Science at Brunel University, London. Today, he'll be discussing on the application of data science techniques in biology. And now, without further ado, I would request Sarat to start with this session. Over to you, Sarat, and thank you for joining us today.
Thank you very much for the invite, which allows me to share a bit of my research. Um, Today, first of all, good afternoon, and I hope everyone and your family members are doing well in these tough times. Uh, in this session, what I would like to focus on is how we can apply data science techniques in biology. So, we live in a very nice world. It has many elements. We In a non-pandemic time, we roam around the world. We do travel a lot. We have governments. We have industries. We need to educate ourselves. We need to do a lot of research, and all of this activity generates a lot of data. And hopefully, in future, we will also be allowed not just to travel across the planet, but maybe in space. And of course, that requires a lot of advancement of science. So, how do we understand this world? We understand this world in form of numbers, and another way of representation is strings, as we programmers like to call them. So either it is through characters or through numbers. And this information uh, is very dear to us when it is in clear structured format. We are really lucky if we get it in that representation. But um, it also has a step of that's why a step of data cleaning and formatting. But let us assume that we get this data in form of um, clear structure with no missing values. So we understand the world, again, to reiterate, in forms of numbers and characters, and somehow we need to understand it. And how do we understand it is, again, we represent it through characters and numbers. We run analysis, apply some mathematical techniques. Sometimes we call it statistics, um, represent it through graphs. And then slowly we are able to build a model of the world. Hopefully, the assumption is that the model we have actually built is very much resembles the world we actually started with. And if not, then there is something wrong with either the input data or the model we have extracted from our input data. So either way, this is a very important step, that is extraction of data. Either we are not um, doing the right kind of observations, getting the right numbers. Assuming that we have that, we have to create a good model that actually represents our world. So this is the basic fundamental of science, that is, to understand something, we make some observations, we apply some statistical or mathematical or other applied science techniques, and then we need to build back the world from which we get this data, and hopefully it matches the data, the world from which we got this data. And if it does not, we repeat this process until we improve our understanding. I hope some of you keep a track of this website and look at what are the emerging technologies, and based on analysis of things that are happening in 2020, um, self-supervised learning, generative adversarial networks, um, DNA-based chips, a lot of explainable AI are some of the emerging technologies that hopefully will ease our life and also help us explain how the world is to us. What is the big problem with all this incoming data is all of this, we are getting tons of data. Tons is an underrepresentation of it. We are getting zettabytes of data, which requires a lot of storage. Not only just storage, then we need to build clever algorithms for analysis. We need to transfer this data around, and then we need to have clever algorithms to query this data. And then it also raises the question of how do we actually ethically treat this data. I highlight it in red because that's a very important piece of information. For example, we really don't want Twitter to share all our personal data, though it shares our thoughts with the entire world without our permission. So it is very important how we ethically treat this data. So, and we store this data in large data centers or in local data storage servers. Now, how does this translate to biology? Well, how can we represent biology? Or let us take example of a human being. Human being can be understood, at least that's the assumption, that we can understand a human being from uh, their DNA. What is DNA? DNA is a large molecule which consists of several billion nucleotides. How can they be represented? They can be either represented as three-dimensional structures, which I will introduce to later on. But there is another way of representing data. I told you about numbers, but we can also represent a DNA as set of characters. And though when you look at it first, it looks quite meaningless, but it is a series of characters that contains a lot of information. So what kind of information does DNA store? So you have the DNA, and then this DNA codes for, that is, it tells the cell what kind of proteins a particular cell has to produce for the survival of the organism. So how many proteins do we have in the human body? So each cell in our human body can generate from anywhere between 80,000 to 400,000 protein molecules. Each of these protein molecule is different. It has a different biological function. Why do we need to understand them? Well, to understand how an organism actually survives, we need to understand how proteins actually perform all the necessary biological functions inside the cell to keep the organism alive. So we are all now familiar with the SARS CoV-2 virus. In contrast to human beings, it has only 29 proteins, which it requires all of those 29 proteins uh, to carry out its life purpose, that is simply replicate itself in a host organism. Unfortunately, it is us. So how can we represent proteins? You might, if you have not done any uh, biology class, you might think, hey, this is something that might be squishy, or something related to dissection, or something else. It is nothing like that. You can understand a protein again through a set of characters. That would be its sequence. And then, which is one way of understanding a protein. The reality is that this protein actually has a three-dimensional structure. There is a way of representing this three-dimensional structure in ASCII format. And this three-dimensional structure is actually responsible for the biological function. One of the examples would be a catalysis reaction. That is, when you actually have some food, it needs to be converted into glucose, and that glucose has to be processed to generate energy for you. And that is actually done by one particular protein called triphosphate isomerase. So that is basically a reaction where glucose, through series of actually not just one, but 10 proteins, is converted into ATP molecule, which gives us energy. So we have thousands of proteins, and these proteins are involved in multitude of functions. I told you that these three-dimensional entities are proteins. They can be represented in ASCII format. ASCII format, for those of you who are not coming from non-programming background, is simply a human readable, editable file. Word document is not in ASCII format, because if you try to just simply open it without the Microsoft processor, you will see gibberish text. Whereas if you open it, this kind of file, which is kind of a PDB file, through normal text pad, then you will see a human readable content. So that's very important. File formats are very important. They dictate how the data is stored, how much storage space it will take, and what kind of a program you have to write to extract data from it. So how can you represent this protein structure? So protein has several 100 to 1,000 atoms. Each atom, for example, the most common atoms in a protein are hydrogens, carbon, nitrogen, and oxygen. And what you see here is there are different elements in this file format. One of the elements, if you look at the atom lines, you will see that it has an index, number 1, followed by N, representing that it is a nitrogen atom. What follows after represents the amino acid. Doesn't matter much for our discussion, but what is important are the three-dimensional coordinates, that is the XYZ coordinates, specifying the position of this nitrogen atom in a three-dimensional plane. That's very, very important. And you have these XYZ coordinates for all the atoms that are present in the protein. So this file can be read by a software called PyMol or VMD, which are visualizing software, graphics software, and they convert this file into that nice blue structure on the left. So this is how we can understand proteins that are present in all the cells in our body or in any other organism of our interest. A little bit of facts. So the first person to actually figure out how to get the three-dimensional coordinates of a protein using a technique called X-ray crystallography was Sir John Kendrew. He was awarded a Nobel Prize for this in 1962. There are different techniques that you can use to solve the three-dimensional structures of protein. And what you see here is a protein called myoglobin. And how many structures do we have so far in publicly available databases is close to 200,000. The number of structures being deposited is increasing exponentially through different techniques. What are these techniques? Not important, but this data is available to us, to everybody. This is open source data. That's again something very important. Open source means data that is freely accessible to anyone, and it is not behind any kind of paywall. And what is the size of these proteins? These are the size of the proteins can vary from few nanometers, that is the unit of measurement that we use, containing few hundred residues, to few hundred thousand residues, which can be a very large protein like a ribosome molecule, shown in pink. It looks very complex, but again, you can represent it in the three ASCII format that I have shown you earlier. These proteins, they exist in different conformations. That is, over time, uh, atoms start moving around, and because of a change in position of a single atom, it results in a new conformation which has a different energy value. And the neat thing is you can calculate what is the probability of a particular conformation based on a particular energy, uh, a- um, uh, a- having a particular energy value. Um, uh, all of this comes from statistical thermodynamics. Um, and all of this is very important to understand how proteins actually work. So what I have told you is that atoms can move. Oh, how fast do they move? They can move at different time scales, from femtosecond time scales. This is something that we have never experienced, but atomic motions occur at femtosecond time scales. To let's say folding of an entire protein, it occurs at um, time scales in seconds to few hours. And we have different techniques that can look at different time windows and tell us about how these proteins work. So experimentally, we can gather information on events that are occurring at different time scales for proteins, but then we also have theoretical simulations, computational simulations that will tell us uh, again the same information. And this is how with theory and experiment we can better understand how the biological function um, performed by a particular protein takes place in an organism. So that's very important. So theory and experiment go hand in hand, and that's where computational techniques come into picture. So what is the problem? A protein can exist in infinite number of conformations, close to, I'm calling it infinite because I cannot really count the number of conformations that can exist. Experimentally, we can access or see a few of these conformations, but in actual reality, we have um, way too many conformations, more than we can count. So coming back to again, what is the challenge? Let's say a single confirmation can be represented in one file, and the format of the file, it has 9 to 10 columns, um, and few thousand lines. Then I have told you that in reality we have, let's say, 10 to the power of 12 confirmations. Theoretically, using computational techniques, we are only able to access few million confirmations. So if a single file, you need a million files to actually understand the physics of how a protein works. And each file, if it takes few megabytes of space, you end up with few gigabytes of storage. Um, and how long does it take to generate all these confirmations? Well, you need specialized computational facilities like Archer, Jade. In India, we have CDAC Param in Pune with a few thousand. I think CDAC Pune at that time had around 6,000 or so processors. Um, and you take around, let's say, 4 to 10 CPUs and run the simulation for 24 hours to 1 week, and then you end up with a million structures, and that's 10 gigabytes of data. Is that enough? If you ask me, I would say no. For a single protein, for statistically reliable results, you need to have multiple replicas of these simulations. That is, you need um, not just a million, let's say, but actually at least 10 million structures. So that translates to 100 gigabytes of data uh, to start building your model and understand something about the biology of this. To recap, what did we learn? So a protein can exist in multiple conformations. Each conformation can be, let's say, represented by C, has a particular energy value E. Energy of all conformations is not the same, which is something we refer to as degeneracy. And experimentally, we may not be able to access the conformation that actually contributes to the biological function. Not always, but uh, that is what is discussed in the field of structural biology as hidden functional states. They are called as dark states. Maybe a bit of a reference to Star Wars, but so here on the right, just to show you a bit of clarity, I have shown you a protein called triphosphate isomerase, which I was telling you about, which is required for processing of glucose, which exists in two conformations, open conformation and closed conformation, which is based on the blue and green loopy structure. So that is the change in the atoms over there. And you might wonder, hey, well, this simple change, does that is that really significant? Let me tell you, change in few atoms over there totally destroys this conformational change if precisely done, and that can lead to the death of the cell and death of the animal. So um, it's very crucial we understand how this protein works. And of course, people have been at it for a long time. So let me try to put it in a different context. So here I have some movie posters. In my childhood, when I used to walk around or go around the city, you used to see many movie posters. And from that movie poster, you have to decide whether you are going to spend your money to actually pick a movie and see if you are going to be entertained for 2 to 3 hours. So exactly looking at a single structure and then deciding what the biological function is going to be is like picking which movie is going to be good just based on a single poster. It's going to be I think you will definitely understand that not all movies represented over here are good. And just predicting that from looking at a single movie poster is very, very difficult. How about if I give you more than a few movie posters? Can you still tell me what is going on in the entire movie? I don't think so. At least just based on all the movie posters of Interstellar, I couldn't predict, spoiler alert, that Matt Damon was in this movie and he had a critical role to play in it. I'm very sorry if I spoiled this movie for you by revealing that Matt Damon is in that movie, but that movie came out long ago, and if you have still not seen it, I can't really help you. But go and watch it. So, coming back to proteins, just based on a single structure, trying to tell biological function is very difficult. So that's why you need theoretical data along with experimental data to understand the biological function. And how can you do that? So first, you need to build a very nice model of how atoms in a protein actually interact. The physics of this, I'm not going to go over it in this presentation. I just added the slide for um, uh, physics enthusiasts. So we have some equations going in which describe the interaction between all the atoms that are present in a protein. And then we come back to nice file formats. So what do you need to describe? For each atom in the protein, you need to describe what is its mass, what is the partial charge on each atom. So we represent this as a topology file. And then we need to describe the connectivity between all the atoms. That is done through few more files. You see, all these files simply either contain some numbers or some characters. So data science, basically. So one of these lines is actually telling you, for example, let me go to the next slide. I think that's where yeah. So for example, here, the highlighted line is actually telling you that the 18th atom is a carbon and the 19th atom is an oxygen. And in the connectivity file, it is telling you that these two atoms are actually connected by a chemical bond. And then how can you to solve that equation over in on the left, you need three values. That is the equilibrium bond distance, that is the r naught value that is present in the parameter file, that is shown as 0.12290, and you need the kij value that is also present in the bond file, that is the parameters file, which is 476 976 kilojoules per mole, and you get the Rij value from the PDB file. And over the course of the simulation, this Rij value keeps changing, and then you can actually calculate what is the potential energy of this bond. So how do we run the simulation? The starting coordinates, I told you, we get from the PDB file. Uh, we had nice potential energy function from which you can calculate the interaction energy matrix over all the atoms. And then from that, based on Newtonian dynamics, we can extract the forces between all the atoms. And from the forces, you can kind of predict what would be the how the atoms would move in short amount of time, roughly speaking. And you can save those coordinates. You can repeat this cycle and generate your million structures. All of this is done on either CDAC Param or in UK we have Archer 2 supercomputing facility. And then now we come to the data analysis part. So how do we analyze this data? So I told you that a protein is a set of few thousand atoms. Each of them can be represented by a sphere. So I told you at time t delta t, at time t, that is the configuration of the atoms. And after a small time step, that is delta t, you might have noticed in this picture that the location of the blue spheres and the orange spheres has changed a little bit. And then again at n delta t time, again the atoms have moved around a bit. So you can monitor these distances. So how do you do this? So you have to write or use existing piece of software to calculate the distances and the distance matrices between these atoms. And then you can also extract the interaction energy between these atoms. So now you have these two variables as vectors. What can you do? You can do exploratory data analysis. That is, you can calculate all sorts of neat statistical properties, mean, median, mode, calculate how the distance distributions look like, whether they are unimodal distribution, whether they are bimodal distribution, whether your graph is shifted a little bit to the left or a little bit to the right. And then you would like to know which distances have actually significantly changed or have not changed at all by employing statistical analysis methods. And then you may not want to look at all the atoms because let's say you have 1,000 atoms in your protein simulation. If you calculate a distance matrix, assuming that you are looking at only half the matrix, let's say I'm not even going to try to crunch that number. Let's say you are looking at 500,000 interactions. You not all the interactions would make sense, so you want to reduce the dimensionality of the data. So you want to employ dimensionality reduction methods. And then after all this, you might be actually interested how on identifying, you have a million structures. How many of these structures are actually unique? Because in the end, all the million structures might look exactly the same. So you have to employ different type of clustering methods um, to identify how many type of structures you have in your data set. You can also employ Markov state models to identify, let's say you have identified 3 or 4 unique structures. You want to now understand how does this protein change between these 4 structures over the course of my simulation. So then you can employ Markov state model, calculate a probability transition matrix, and from that you can calculate all sorts of interesting things like transition rates, free energies, so all sorts of interesting properties that you can compare with experimental observables. And then you can write neat papers and maybe one day get a Nobel Prize. That's the idea. So how do we do this? We write either our own code, like a small code over there shown in Python, if some of you have recognized that, or we can use R for exactly this purpose. The language doesn't really matter. You can use Java. I would not recommend it for this purpose, but R or Python is something that we would like to teach. Um, in our masters, we focus on R. You also learn Python, and then during this journey, you will be employing different libraries coming either from, again, from Python or R. Um, then after you have developed your software, you would like to put it up on places like GitHub so that you can showcase it to your employers, to show how your cool programming skills, or share your cool software with the rest of the world. Uh, One example that I would really like to give you shortly is about progress made by DeepMind in the field of structural biology. That is, they came up with an artificial intelligence software called AlphaFold that could predict a protein structure just based on sequence. We have been at it, um, doesn't mean just me, but since 50 years or more, a lot of scientists coming from mathematics, physics, and chemistry have all tried to predict what would be a structure of a protein just from the sequence. And many people have made a lot of progress, but DeepMind have come up with a software, and we have to yet look at the details, but with very high accuracy. It's going to be really a landmark event in the field of structural biology. If that method works for a lot of the proteins, at least it does solve, even if it does really well for few proteins. It's a big landmark event. I encourage you to look at this particular blog post, and hopefully in future the article whenever it comes out. Then why do we need all this? One of the problems that you can, if at all you want to think about, is something that you might be aware nowadays is about antibodies. Antibodies are proteins. We have a very good representation over here in that picture, where all these antibodies, I think, are attacking a particular cell or a virus particle. I can't remember which one is that, but these antibody particles help our body identify a foreign pathogen in our bloodstream, and then so that our immune system can actually take care of it. And for that, you need to have antibodies. And for that purpose, we generally get vaccinated so that we are prepared in advance. And how many unique antibodies do we have? We have around 10 to the power of 16 to 10 to the power of 18 unique antibodies based on a recent estimate. And these antibodies are proteins. So if you want to understand how proteins actually work, you need to do experimental and theoretical studies on these antibodies. And given the number of unique antibodies we have, uh, it is going to take a long time unless we have a general principle of how they actually work. So we have a big problem at our hand to apply data science techniques to better our life. And with that, I hope I have convinced you that eh, there is a lot you can do with data science techniques in biology, and hopefully that has generated some interest for you. And with that, I would like to stop for some questions, any questions that you might have. Thank you.
Hi, Sharath.
Hi.
Hi. Sorry, I mean, I'm facing network issues. I just wanted to check. Are you continuing with the sessions, or
So I'm done with my talk, and I'm just waiting if there are any questions.
all right? I do not see any questions in the chat box. Can you please confirm if you received any direct message privately with any questions?
None so far. I just received a hi. There was
Okay.
a question after it.
Okay, fair enough. So I think there are no questions. So what we can do is probably we can just probably end the session, and if at all there are any questions, we will definitely divert it to you. Does that help?
Thank you very much. So if there are any questions, I'm very happy to answer any emails. So thank you.
Thank you so much, Sharath.